Hello, ladies and gentlemen. Welcome to Research Center for Ecology, National Research and Innovation Agency. We call BRIN, and this is our event, ExpertTalks 2. Miss Dila, you can open it.
Yes, Mbak Linda, I hear you.
Okay.
Good morning in Netherlands and good afternoon in Indonesia. We will begin this talk in just a few minutes. While we wait for other participants to join the room, please make sure your audio and video settings are working properly. We also highly encourage you to rename your Zoom account with the format name _ institution and setting up the virtual background that has been provided. Thank you, and we will begin shortly. Before we begin with our formal agenda and have a productive session today, please allow me to read a few guidelines for today's session. First, please ensure your microphone is muted throughout the presentation to avoid any background noise. Second, if you have any question during the session, feel free to drop your question in the chat box using format name, institution, and question. Or if you have any question that you're willing to convey directly to the speaker, you can use the raise hand feature during the Q&A session. Please be respectful and concise with when asking question. And then group photo will be taken after the speaker's presentation and will be led by the moderator. Lastly, the link for the attendance and e-certificate will be shared at the end of the event. Okay, shall we begin our agenda for today? Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning in Netherlands and good afternoon in Western Indonesia time. Distinguished guests, researchers, ladies and gentlemen, Bapak dan Ibu, welcome to Expert Talk second series, proudly organized by the Research Center for Ecology, National Research and Innovation Agency, Badan Riset dan Inovasi Nasional, or BRIN. We are delighted to have you join us today, Thursday, June 18, 2026 at 1 p.m. Indonesia time for this scientific discussion on the challenges facing coral reef ecosystems under nutrient pollution. As the introduction for today's agenda, firstly, we will hear a welcome speech from Asep Hidayat, PhD, Head of the Research Center for Ecology at BRIN, and then followed by discussion that will be guided by Dr. Ludi Parwadani Aji, Senior Researcher at the Research Center for Ecology BRIN, who will serve as today's moderator. Following that, the main narration will be delivered by Dr. Lisa Becking, Senior Researcher at Naturalis Biodiversity Center and Associate Professor at Wageningen University and Research. And I am your Master of Ceremony today, Dila Swestiani, Junior Researcher at the Research Center for Ecology, BRIN. Without further ado, let us warmly welcome Asep Hidayat, PhD, to deliver his opening remarks. Bapak Asep Hidayat, PhD, the stage is yours.
Baik, terima kasih Mbak Adila. Thank you for the opportunity. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Selamat siang di waktu Indonesia dan selamat pagi di Belanda dan salam sejahtera untuk kita semua. Om Swastiastu, Namo Buddhaya, salam kebajikan Bapak Ibu sekalian. So on behalf of the Research Center for Ecology, BRIN, it's my great pleasure to welcome all of you to the second Expert Talk of 2026. This event is part of our regular scientific discussion program to strengthen collaboration, exchange knowledge, and promote innovation and ecological and environmental research. Today, we are honored to host an important discussion with the title Coral Reef Under Nutrient Pollution. We are very privileged to welcome our speakers, Dr. Lisa Baking, senior researcher at Naturalist Biodiversity Center, and associate professor at Wageningen University and Research. This session also later will be moderated by Dr. Ludi Parwadani. He is researcher of the Research Center for Ecology Brain. Ladies and gentlemen, college, researcher, student, and participant, it is my great pleasure to welcome all of you today in ExpertTalks. This topic is highly relevant, especially for Indonesia, as the country known as one of the world center of the marine biodiversity. Indonesia is a home for to approximately 51,000 square kilometers of the coral reef, representing around 18% of the world coral reef area. This reef provide habitat for thousands of the marine species and support the livelihood of the millions of the people through fisheries, tourism, coastal protection, and other ecosystem services. However, coral reef ecosystems are facing increasing pressure from multiple threats, including climate change, coral bleaching, overfishing, destructive fishing practices, coastal development, sedimentation, marine pollution, and nutrient enrichment. Among these threats, nutrient pollution has received growing attention from the scientists worldwide. Nutrients such as nitrogen and phosphorus are essential from life in the ocean. However, when excessive amounts enter the coastal water through agriculture runoff, domestic waterways, industrial discharge, and land use changes, they can seriously disrupt the balances of the coral reef ecosystem. High nutrition concentration can stimulate excessive algae growth. These algae compete with the coral for space, sunlight, and resource. Nutrient enrichment can also reduce water quality, increase disease susceptibility in the coral, and weaken the resilience on reef ecosystem against another stressor such as rising sea temperature. Over time, reefs that are dominated by healthy coral must shift to algae-dominated ecosystem, resulting in the biodiversity loss and reduction ecosystem function. Ladies and gentlemen, the question before us is, what can be done? From a policy perspective, governments need to strengthen integrated coastal management, improving water treatment system, reduce nutrition runoff for agriculture, protect critical coastal habitat, enhance long-term monitoring program. At the same time, science and researchers have a crucial role to play. We need more research to better understand, such as the resource and pathway nutrient pollution in coastal ecosystem. The interaction between nutrient pollution and climate change, threshold level of the nutrient loading that coral reef can be tolerated. Innovative approach for nutrient reduction and ecosystem restoration. The use of remote sensing, environmental DNA, and artificial intelligence for coral reef monitoring. Community-based approaches to support reef conservation and sustainable coastal management. Especially for Indonesia, strengthening coral reef research is not only important for biodiversity conservation, but also for food security, climate resilience, and the sustainable blue economy. Ladies and gentlemen, we are very fortunate today to have with us on our outstanding scientists and experts in the marine biodiversity and coral ecology. Dr. Lisa Becking has intensive experience in the coral reef research, marine conservation, biodiversity assessment, and ecological monitoring. We are honored to learn from her expertise and insight on how nutrient pollution impact the coral reef ecosystem and what lessons can be applied to improve coral reef management in Indonesia and beyond. I would also like to express my sincere appreciation to all participants who have joined us today. Your presence reflects our shared commitment to advancing ecological knowledge and supporting evidence-based environmental management. I hope today's expert talk will inspire new ideas, foster future collaboration between researchers, institutions, and also countries, and strengthen our collective effort to protect coral reef ecosystem for the future generation. Finally, I would like to express our sincere appreciation to speaker, organizer, moderator, and participant who have joined this event today. I wish all participants a fruitful and inspiring discussion. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh. And then I give time
Waalaikumsalam.
to Badila.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Bapak Asep Hidayat, PhD, for the warm welcome and inspiring words. And then, ladies and gentlemen, Bapak dan Ibu, we now arrive at the core part of this webinar. This main session will be fully guided by our esteemed moderator, Bapak Dr. Ludi Parwadani, Senior Researcher at the Research Center for Ecology, BRIN. Without further delay, please welcome Dr. Ludi Parwadani. Dr. Ludi, please take over.
Thank you, Mbak Dila. Good afternoon to everyone joining us from Indonesia, and good morning also to our guest speaker, Dr. Lisa Becking. Welcome to today's expert talk on Thursday, 18th of June, 2026. So it is currently around 8 EM in the Netherlands, so we would like to extend our sincere appreciation to Dr. Lisa for taking the time to share your expertise and experience with us today. This expert talk is attended primarily by researchers and staff from the Research Center for Ecology of BRIN, and we are delighted to have this opportunity to learn from you, Dr. Lisa Becking. We would also like to express our gratitude to Pak Asep Hidayat, PhD, as our head of the Research Center for Ecology BRIN, as well as all members of the organizing committee like Ibu Linda Wikeningrum, Mbak Dila, and for making this event possible. Before we begin, it is my pleasure to give a short background of our speaker, Dr. Lisa Becking, who will deliver a presentation entitled Coral Reef Under Nutrient Pollution. Dr. Lisa Becking is a marine biology senior researcher at Naturalist Biodiversity Center and associate professor at Wageningen University and Research. Her research focuses on understanding how marine biodiversity originates and responds to environmental change, with a particular emphasis on tropical marine ecosystems such as coral reef and marine lakes. With extensive field experience in Indonesia and the Dutch Caribbean, Dr. Lisa combined ecology, genomics, biodiversity surveys, and conservation science to investigate the resilience of marine ecosystem under increasing pressure from climate change, pollution, and human activity. Her work contributes to evidence-based strategies for marine conservation and sustainable management of ocean resources. She leads and collaborates on international research initiatives, including projects on marine biodiversity change and conservation. She is also an educator and mentor, supervising graduate researchers and teaching marine ecology at Wageningen University. She regularly speaks to academic, professional, and public audience about marine biodiversity, coral reef resilience, sustainable tourism, and the future of our ocean. Through her research and public engagement, she seeks to bridge scientific discovery and societal action to protect marine life for future generations. Okay, Lisa, time is yours. I give back to Mbak
Good
Dila.
morning. Oh, sorry.
Oh yeah, sorry. Time is yours, Lisa.
Oh, okay. Sorry. So, good morning from Amsterdam and selamat siang in Indonesia. So, thank you so much for inviting me and giving this opportunity to speak. I particularly would like to thank Dr. Asap Hidayat for inviting me and hosting me here, and also for the kind introduction, as well as Dr. Ludiaji for inviting me and giving the introduction. And it's great that there's so many participants, and that so many people are joining from the Research Center of Ecology from BRIN, and I guess also from other research centers within BRIN. So I will share my screen. Hold on. Can I just confirm that you can see my screen, Ludi?
Yes, we can see your screen, Lisa.
Okay. Great. So what I want to do today is I want to talk about nutrient pollution in coral reefs, and what I will do is I will give a little bit of an overview of the different programs and projects that we do, together with a large amount of partners, as you can see below. And there are different studies that encompass different parts of measuring, monitoring, and managing what we think is a bit of a hidden stressor to biodiversity. It's a very important local stressor, but it often gets overlooked. And so that's why I want to, for this session, just really focus on these. There are, of course, other stressors, but these are ones that we think are very important, and also, importantly, you can manage them. This is a stressor that actually can be managed and mitigated. Okay, so I will start off with talking about the InReef program. This is a 6-year program focused on, it's an interdisciplinary, or actually transdisciplinary program, to build the resilience of marine protected areas and tourism destinations. And so, as was already indicated in the introduction, coral reefs support, the livelihoods, food security, and coastal protection for millions of people worldwide, and definitely also for large portions of the populations in Indonesia. However, a lot of the coral reefs are changing. They're changing their biodiversity, and there are many reefs that are shifting from coral dominance to dominance of other organisms. For example, here you have cyanobacterial mats. They're the red mats here, which are toxic for corals, and also create anoxia and kind of reduce the resilience of coral reefs. And we see this particularly in areas that have high nutrient pollution. But so we know from various studies, and you probably also have heard, that there is generally quite a decline in coral reefs, in corals. However, I think it is important to stress there are still a lot of coral reefs around, and definitely in Indonesia and the Coral Triangle. And what is there, we should really protect and also celebrate, I think. But let's talk about those sources of decline. I think you can put them in two areas. You have the global stressors of climate change, and particularly for coral reefs, the key problem is increased seawater temperature. And then you have many local problems, many local stressors that stress the reef, that reduce the resilience of the reefs, and primary among those are overfishing and low water quality, or pollution, water pollution. And think of things of coastal development, erosion, runoff from all kinds of things from land, and untreated sewage. Now, what we've been focusing on in our team is really this aspect of low water quality and water pollution, because we feel that overfishing is super important, but there is a lot of attention, and a lot of people are working on that, which I hope they keep doing. But we're very interested in, like, what is this sort of hidden or invisible stressor? What is it doing, and what can we do to prevent it? And just to give a little bit of an idea, so basically what you have is that you have increased sedimentation and runoff from agriculture or sewage. It creates nutrient enrichment, so increase of nutrients in the coral reefs, which ultimately leads to what we call eutrophication. So that's when you have such a high increase of nutrients that the whole biodiversity shifts from coral dominated to dominated by microbes or dominated by algae. Because a key thing about coral reefs is that coral reefs in a, let's say, more pristine state or in a more natural state typically are what is called oligotrophic. So it's a system that is very efficient because it normally doesn't have a lot of nutrients. So it's very efficient. All the organisms work together to catch the nutrients and to pass it on. So when you have a situation where you add lots of nutrients, it's kind of like adding lots and lots of junk food. And it's not the corals that are growing, but it's the bacteria and the algae. So you really have a change in system. Now, but one thing that I wanna stress again is that there's a lot of there's a lot of news about the declining coral reefs, as I was saying before, and there are problems, and it's very good that there's alarm. However, what gives us hope is that there's more and more research that shows that if you can reduce the local stressors and really reduce the local stressors, you increase the resilience of the ecosystems. And so in the case of coral reefs, it means that local management of local stressors really does buy us time and makes the coral reef stronger and more resilient towards the effects of climate change. So we need to deal with climate change, but on a local level, we can also do a lot. Now, one source of local change, like a big source of local change, is tourism. And tourism takes on lots of different shapes and forms. As you can see here, you have cruise ships, you have homestays, you have different forms. And they all come with different ways of stressors and different issues. And one of the things that we're interested in is a little bit this double-edged sword that tourism presents. Because on the one hand, tourism can really, it can support the local economy, it can finance conservation, it can support conservation in many ways. But if tourism is not regulated and if it's just left unchecked, it typically, runs out of control, and it can really become a stress both to the ecosystems as well as the societies in economies above water. And just to give a sense of how tourism can influence this nutrient pollution, many tourism locations do not have sewage treatments. So that means and this is an example from Aruba. This is an island in the Caribbean. What you're seeing here, this brown on the left, this is after raining. This is untreated sewage just going straight into the coral reefs. And we see this in many different locations, also in Indonesia. Locations with high tourism areas they build the hotels, they build the roads and the boats, but no sewage treatment. So it means that untreated sewage is going straight into the water, which is not good for the coral reefs, but it's also not good for public health. And so on the one hand, this is dramatic, but on the other hand, we know what we need to do to tackle it. It's dealing with coastal, building adequate sewage treatment systems, and preventing erosion and preventing development on the coast. So these are the solutions we know, and they're related to public policy, public government decisions, as well as private parties. Because tourism, it has various effects, but again, also these cruise tourists in the Caribbean, and you also see that this is increasing also in Indonesia. These cruises have 3,000 people on board, which means that's a lot of people going to the toilet. And so there's this one group, they estimated that if you go on a cruise for a week, there's about 200,000 gallons of sewage, which in weight would be the same as more than 200 elephants. And all of this gets dumped in the sea. So this has effects on the systems. Now, my team and I have been working in Indonesia for quite a while, and we've been working in different marine protected areas. So these are areas that have been identified as high conservation areas, and then there's an area that's been designated as either a national park or a marine protected area under the Ministry of Fisheries and Marine Science. And typically these locations are beautiful, like this. This is a drone image from Miso in West Papua, and they're typically beautiful, and as such, they're typically highly promoted for tourism. Also with the idea that tourism will bring in revenue for the parks through permitting or otherwise. However, what we noticed is that even though they're promoted for tourism, there's actually very little regulation and governance for tourism. So it's just let loose. And what we've learned from the Caribbean, from Bali, from other areas, is that if you don't regulate tourism, it just grows. So based on that, we set up with partners in Indonesia, we set up this in-reef program where the aim is really sort of trying to look at questions of how do you balance tourism in marine protected areas in order to safeguard both the ecosystems underwater as well as the societies above water. Now, this program is running. It will run in towards the end of 2028. And the aim is to identify and develop tools to analyze how the resilience of marine protected areas can be measured, monitored, and managed. And there are 10 PhDs that are part of this, and a postdoc. And what it's quite a transdisciplinary team, and all the PhDs are either from Indonesia or from the Caribbean. So for us, it was very important that the PhD candidates are from the countries where the research is being done. And so what we have PhDs that are focused on, let's say, the biophysical characteristics, so let's say the hydrodynamics, and these things, as well as the biodiversity. So they're kind of disciplinary, also on pollution and tourism governance and socioeconomics. But then we try to link them all through these topic lines to make certain that they also interact, and that we get sort of interdisciplinary work. So through topic lines of marine protected areas, as well as indicators, where we're also looking at socio-ecological systems and adaptive management. And central throughout it all is an education program, and I'll talk about that in a moment. So where we're working, so this is an image from Spalding, showing sort of the epicenter of marine biodiversity, where Indonesia is right in the middle. So we're working there, as well as in the coral reefs of the Dutch Caribbean. And then together with partners on the ground, we selected marine protected areas that are that are sort of on a gradient of tourism development and reef health. And based on that, we selected these different marine protected areas, but there are a couple of them that we're really focusing on, where we do most of our work, and then we use the other MPAs as like case studies or test sites. Now, our approach, which sort of makes certain that all these different disciplines interact, is by taking a socio-ecological systems approach. And the idea being that a marine protected area, it's an ecosystem, but it's also a social system, and it's arranged by governance arrangements. And as such, it's a dynamic social ecological system. And subsequently, the MPA is very clearly defined, has very clear borders, but it's ecologically, physically, economically connected to the world outside. So we're also looking very much, the key question that we're asking is, how do these outside flows influence the dynamics in the marine protected areas? And how do the dynamics within the MPAs, how do they interact? How do they interact? That's one. And then also what are areas where you could potentially do something to increase the resilience of the MPAs? And with resilience here, I mean very much from the perspective that the MPAs can deal with shocks, and that's because there will be shocks. There will be either economic shocks or from climate change, but that that the system doesn't collapse, but that it can come back. And so what we really wanted to do was that where I think our novelty comes is that we're not just looking at one system, but really looking at multiple systems and how they interact. So we're looking at systems underwater, of the biodiversity and the hydrodynamics, as well as the land-sea interface and on top of the water, as well as, of course, on land. And really with these questions, what are the socio-ecological systems, and then what influence does tourism have on it? And of course, tourism is not one thing. It's also very much the question, the different types of tourism. And so we're aiming to build a MPA resilience dashboard that provides sort of transparency of the status of all these MPAs, and also can be like an educational or decision support tool. Linked with that, with the social scientists, we're working on governance toolbox, but of course that's together very much with the local partners that we're working with who are related with the government. We're trying to set up monitoring guidebooks and training materials, working from what exists and what people are doing, and then just trying to make a lot of things more unified, particularly in relation to water quality and indicators. And then we've taken a very specific issue, namely wastewater, so sanitation solutions, because we see also in these very remote areas that as tourism grows, you just get lots more toilets. And so we were thinking of like, how can you come up with a wastewater solution that's low tech, low cost to reduce this stress from toilets in, for example, areas like Raja Ampat, where you have an increase of homestays and resorts. So then, as I said, education and dissemination is a large part of this project. So we're setting up an education hub. So all the members, both from Indonesia and the Netherlands, are lecturers actually, or teach in some way. We're making knowledge clips. These are online modules. We're hoping to bring those online this year. In any case, for water quality monitoring and for socio-ecological systems explanations. We have fellowships for master students. Yeah, and so this kind of runs throughout. And just to give an example, for example, a workshop, an education session that we had 2 years ago, where different members of the group provide. They gave lectures and also training in the different protocols. We also believe very strongly in giving hands-on analysis, particularly to the younger staff. So we work a lot with NGOs, but Brin was here as well as at universities to really, because what you see is that often a lot of data is collected, but then the analysis kind of stays a bit flat. And so we've been giving a lot of training and scripts on how to do it and how to work on it. And then we also have like online coaching sessions. We've had various feedback sessions in the field, and then also field training in different methods of sampling and water quality monitoring. In this example from work in Bonaire, there we've been working with quite a large team who've been doing long-term water quality monitoring, and they also are working on science towards policy. And so they were asked to give policy recommendations. But before we did that, what we found important was that first to all the stakeholders, present what's been found, and then also present the recommendations. And then we went through discussion sessions in various forums of, what do you think about these recommendations? Does it ring a bell, or do you think other things? And just to make certain that, because sometimes as a scientist you can come up with these ideas, but at the end of the day it's about who's on the ground and what they're doing, and whether they see that link well or not. And we also give on-site eDNA and DNA barcoding training using the nanopore, this very, very small sequencer. And we've done this also in Indonesia and wish to do this more. So also, if there's interest in this, let me know, and we can talk more about doing this potentially also at Brin. We've been wanting to do it for quite a long time, but it's been, because there have been so many changes in Brin, it's been a little hard to coordinate, but I'm hoping that we can do that next year. And so here's an example of a fellowship. So this is Bema Monim. So he's a practitioner within the monitoring team of the Reef Health monitoring team of Papua University, as well as Yayasan Konservasi Alam Nusantara, YEKAN. And we provided a fellowship so that he can follow the full MSc program at UNIPA, and while for his thesis he's working in Reef. Now, what I'll do now is I'll give some examples of some of the findings that we have. We're still very much in work in progress, but it gives you a bit of an idea of where we're going and other things. So this is some work done by Agustin Capriati, who's a PhD candidate within my team and almost finished. She's also the coordinating researcher of INREEF. And so what she was interested in was, okay, so there are many publications on the stressors on coral reefs. So she brought all of those together, and then she was like, okay, so Indonesia currently has many MPAs, right? I mean, more than 400, and every year more MPAs are being created to get the target of 30 by 45. And then her question was, to what extent are these stressors incorporated in the marine protected management plans? And so she did sort of automated text analysis using Python. That was published last year. Quite nice with our a large team from different organizations in Indonesia. And so what you see here is this is Indonesia, and you have this is, by the way, this is data from 2022. So what she found then is there were about 379 MPAs. Those are across the country. But of those, only 77 of the MPAs had a management plan, and a complete management plan. So that means with a clear introduction, problem statement, zoning system, as well as a action plan. So that already in itself says quite a bit, that a lot of MPAs have been designated, but there's not a full management plan. So there's a lot of work in progress here. To be clear, these are only the MMAF MPA, so these are not the one from the Ministry of Forestry, the Taman Nasional. So this is only for the MMAF. So first off, she found, okay, the majority of the MPAs don't have a management plan. But okay, still, if you look, those gray dots are across the whole country. So that's great. This is a great start. And then she looked at all of these 77 MPAs. And her question was, okay, we have these different stressors that we know, that published, that we know these are stressors on coral reefs. And to what extent are they incorporated in these MPAs? And the way you can look at this is that you have these different colors. So you have red for fishing activities, green for land-based, yellow, orange for climate change, and blue for others. And then she looked at, okay, to what extent are these terms? And then these terms, they're here in English, but what she did is she used a composite of different terms in Bahasa. And then for example, here you see that blast fishing, cyanide fishing, and destructive fishing are very prevalent within the text. So they're some are only in the introduction, but then they're not in the zoning. Some are only in the zoning, some are only in the actions. And then what the ideal situation would be is if that stressor is mentioned in all of the different segments of the MPA management plan. So you see that a lot of them are focused on fishing activities, which makes sense because I think a lot of the MPAs originally were very much created to, protect fish stocks, and ultimately with the idea that the fish stocks would grow and it would increase. So then we also see, which is good, that sand mining and deforestation is also identified a lot. Climate change to some extent, the way you should look at it is, about half the MPAs mention climate change, but that also means that about half the MPAs don't mention climate change at all and don't mention bleaching at all. So it is something that's sort of identified, but clearly not a lot. And then what I found surprising, because in the past years, I think it's been great how Indonesia's been really much more aware and vocal in their many activities on plastics, and plastic pollution and waste. But actually, it's not in the marine protected management plans. Most plans, so I would say, over 80% of the plans don't actually address plastic pollution. Also, there's some addressing mineral mining, but not a lot. While this is a key risk for marine protected areas. And then, the topic that we're working on, actually, the majority of marine protected areas don't address nutrient pollution. So either in forms of eutrophication and or coastal development. So what she wanted to do then is she really looked at, okay, so for example, destructive fishing. It's identified in a lot of the management plans, and it's identified as a problem in the introduction. Then the question is, to what extent is it addressed in the zoning? So then she looks at the overlap to what which MPAs actually have addressed it in the zoning, how many then actually have it in the actions. And for example, here, if you look at those numbers at this Venn diagram, you see that actually only 4 plans of the 77 address destructive fishing in the intro, in the zoning, in the action. So that means that there's actually something going to be done. because you can identify the problem in an introduction, but then if you don't have an action plan, then it's a bit hard to do anything. And so if you do that, then for, let's say, sewage discharge, you see that very few MPA plans actually address it, and only one has a complete introduction, zoning, and action for it. Climate change, somewhat more, and then you have coral mining, also quite little. So what we can conclude from this is that, nutrient pollution is really not really on the map yet. So nutrient pollution, eutrophication, sedimentation, as it stands now, is not really being addressed. So it's really something we wanted to highlight, because it's something, the way you can see it is, I see that there are many MPAs that still need a plan. There are many new MPAs. So I would say, based on this work that Agustin did, that we have kind of a checklist of things that, you should try to address in your MPA plan. And one of them really is eutrophication and nutrient pollution. So then, okay, we have nutrient pollution, but then the question is, what's your standard? What's your threshold? And that really is a tricky question. So a student from IPB, what she did is she compiled a lot of information from different countries, and then tried to get what the official national threshold is for chlorophyll A, for dissolved inorganic nutrients, nitrogen, and for phosphate for the marine systems. And what we see is that, it's not really a clear picture, because, for example, chlorophyll A, most countries don't have a threshold. Only Australia does. And then for dissolved inorganic nitrogen, you see that the values are all over the place, right? So Indonesia has like 18, but then the Philippines has 163. So and ASEAN Secretary has like 5. So what we then said was, let's go to the science, let's go to the papers, and there are a number of researchers who have worked in the Great Barrier Reef and Guam and American Samoa. So we stuck to, let's say, Pacific and Indo-Pacific systems. And they've identified a couple of thresholds that you kind of want to stay under that for the coral reefs not to be too stressed. Again, it's still sort of a start, but you see some values here on the top table. Then if we look at the table below, that's where we have the values of, let's say, the national standards. And if we look at what Indonesia has for dissolved inorganic nutrients, that is many times higher than what coral reef scientists say is a good idea. So clearly there's work to be done there. Also, and we'll get to that in what we find, is I think it's also maybe a little dangerous to have one threshold value for all of Indonesia. I mean, think about it. Indonesia is huge and it has so many different systems. So probably the threshold values will have to be established per region or per MPA, but having a full national one is going to be hard. So what we then decided to do, and this together with Baban Avaludunir Ahmad from Yekan, together with researchers at IPB as well as BRIN, was sort of like, okay, so there's lots of standard coral reef monitoring happening. And we said, okay, here, take a CTD that also measures chlorophyll A, and every time you do your monitoring, also measure your chlorophyll A, and then also try to get your dissolved inorganic nutrients, just to get a bit of an idea of like, what is the distribution and what kind of values are we finding? Because right now we don't really know. Like, most of the water quality measurements that are done in the marine environment in Indonesia are based on satellites, which is really good, but it's really high resolution. So you're not really getting that land sea dynamic on a smaller scale. So this is from all kinds of monitoring in Lease, Maluku, in Nusa Penida and Bali, in Raja Ampat, in different locations. And this is what we find. So I'm not going to go into every single detail, but what we did is we categorized each location on whether it's like an undeveloped area or urban developed, or more like urban development mangroves, or open water. And then what we have is this is all the chlorophyll A values. And then what you see, what the lines, the dashed lines, are the optimum values or the maximum values based on the science. And then the sort of pink bar are the threshold values set up by ASEAN. And one of the things I found striking was that, if you look at these values, most sites, except for Southeast Misol, are above the threshold values. Now, I want to be very careful with the conclusions here, because I know a lot of these sites, and it's very clean water. There's very little pollution. So this may just mean that this is just ecologically a different kind of system than the Great Barrier Reef. So maybe, for example, in Nusa Penida, there's lots of upwelling. Also in North Misol, it's very deep. So the reason why there's high chlorophyll A may not actually be because of pollution, but it's just that this system just has higher chlorophyll A. So it's very important to get this baseline information correct before you make conclusions about thresholds and whether things are bad or good, let's say. And this is some examples from phosphate from Misol and Dampier. And here too, we see that, well, first off, you see that these thresholds are quite different depending on whether you take the science or other, depending on what thresholds you take. And here too, we see that a lot of the locations are higher than many of the thresholds. And again, I don't know if that's actually because there's pollution or whether it's just that these systems have maybe some higher nutrients. And the other thing that we found is that, of course, nutrients in water are highly dynamic. And so all of this data is just based on sampling once and just in a location wherever the diving is. And so this is work that is almost published. It's in press right now in ecological indicators, led by Agustin together with the Coral Triangle Center at in Ambon. And what they wanted to do was really look at integrating these physiological, chemical, and microbial indicators to assess the small-scale nutrient pollution. And what we were thinking was, because nutrients in water is so dynamic, we're thinking we should also really take into account the distance from land to sea. And so Agustin was working in Lease. It's a recently set up marine protected area in Maluku, just beyond Ambon. And she went to sites that had no development, that had a lot of development, and then areas where rivers come out. And then she went from the coast to the ocean and sampled at 1 meter, 5 meters, 10 meters, 25, etc. And then measured these physiochemical parameters. And that was like total suspended solids, chlorophyll A, all these different things, as well as the inorganic nutrients. And what you found is that the values close to the coast are highly dynamic. So here you see with the chlorophyll A close to the coast. So there, it's quite all over the place, both for populated areas, non-populated areas in the river mouth. But then as you move further away from the coast, it becomes a little bit more stable. So and we see that also with phosphate and nitrate. And so what that tells us is that when you are doing your sampling, it's important to take many samples close to the coast because that's highly dynamic. And then also to make certain to take a few samples further out to kind of get the idea of how it stabilizes. And yeah, so this hopefully informs the sort of monitoring design. And then, yeah, then we also really looked at, okay, so we have all these different variables, so temperature, salinity, and the different dissolved inorganic nutrients. And then how do they vary according to, yeah, whether they're populated, not populated river mouth. The pattern is less clear than you would hope, but I think that that is also why people haven't been looking at nutrients so much, because it's not so straightforward. Because if you want to do it, you have to actually take many samples every 2 weeks and keep doing it, and that's really logistically very hard. So this is, but what we still see, there are some patterns where we see that well, the oxygen levels definitely decrease closer to the river mouths, and we also see that the nitrate and phosphate is actually higher in these populated areas than non-populated areas, and also go beyond the thresholds that have been set by the Indonesian government. So these dashed lines are the thresholds. So I would say, based just on this work, it's clear that getting data from inorganic nutrients in the water is useful, but it's not super straightforward. Another thing that we've noticed is that the values are very, very low, and you need very high-tech equipment to measure those values. And there aren't that many places where you can do that. So this is something that's work in progress. But as a result, we've also been looking at what are other indicators that maybe can incorporate time a little better. And one of those are these benthic cyanobacterial mats. So these are examples of what you can see here, and we're seeing that they're increasing in the coral reefs. And what we did is, or what Agustin did is she collected these mats in these different locations as well, and then sequenced the microbial communities in these benthic cyanobacterial mats with 16S, to then see whether the communities could potentially function as like indicators for the status of the water quality. And what you see a very clear separation. So what you see here is like the red is the river mouth, green is the populated areas, and blue is the non-populated areas. And what you see is that there's a very clear separation in the community composition of these benthic cyanobacterial mats between river and non-populated. Populated. Within the populated, non-populated, it's a little less clear, but the structures are very much influenced by the nutrients. So you see that here in the arrows. You see these are all significant variables that significantly explain the variation in the community structure. So you see that phosphate and ammonia influence the structure. So this is an indication that cyanobacterial mats might be a very nice sort of less time-sensitive indicator. But again, this needs a lot more work. So what we can kind of conclude from this work that we've done so far is that what next steps that are really needed are setting up local references and thresholds. I think having one national value is probably not so informative, but something that can be worked on. What is needed is like regular monitoring, but particularly also sort of optimizing these methods of like what kind of proxies can be used to do regular monitoring. And then within the monitoring to really consider spatial and temporal variation. And so I'm just going to end with a couple of the PhD projects from PhD candidates that are working within IMRIF to give you a bit of an idea of the directions that we're working on besides this work from Agustin. So this is where Riza Saribu, he's a lecturer at IPB, but also now is an LPDP scholar, has a fellowship to do his PhD at at Wageningen as well as at Twente University. And he's really looking at, okay, so you can get a lot of information about water quality from satellite imagery. And so he wants to evaluate how can you optimize that. But then what we were showing is a lot of the dynamics are very local. And so he's trying to develop these tools to monitor coral reefs more with drones with all kinds of hyperspectral or multispectral sensors. And so, yeah, that work he's been doing in LEASA, and he has some various preliminary data, but I just want to put that out there so that if anyone's interested in this kind of thing, that's something we're working towards. And then this is work from Tobia in the Caribbean, where he's because in the Caribbean, for a lot of the islands that are here, there is a lot of data already. So he's trying to bring that data together to then develop more of a socio-ecological perspective, and particularly looking at, okay, so the state of the coral reefs that we see today is a response from the things that happened in the past. So really looking at sort of coral reefs, tourism, and coastal change in the Dutch Caribbean and its effects on the coral reefs. And so where he's, just so you have a bit of a sense where it is, this is the Caribbean. So up top is the United States and Florida, and down below is South America. And he's working on these islands of Aruba and Bonaire. And his aim is really to try to understand the tourism reef dynamics, where he's first looking at very much these historical pressures. So how has land change developed? How have decisions on tourism development, how has that already been, and other activities been in the past? And what possible effects have they had on the coral reefs? And then he's really taking a systems dynamic, really looking at feedbacks between tourism and coral reefs and socio-ecological systems. And then there's a finer chapter, more an ecological side, is really looking, okay, so we know these areas have very high enrichment. Do areas with high nutrient pollution, how do they respond differently to stressors or having grazers or not? So that's sort of a insight experiment. And then the final PhD project that I wanted to identify is from Raymond Malawao. He's a PhD candidate within my team at Naturalis and also at the University of Amsterdam. He's Moluccan and Dutch. And his question is very much about, okay, so we see that coral reefs are changing. However, what does that mean for the functioning of the ecosystems? And he's looking for ways to identify that the coral reefs are changing, that the functioning is changing. And his ideas are very much like, okay, so we see that there are these coral-dominated areas, then we see that there are areas that are dominated by algae or by soft corals or by other organisms. And his question was, what does that do for the metabolic balance? Because when you have a dominance of different organisms, your function changes. And really trying to link the biodiversity patterns with these different metabolic patterns. And he's doing that by making 3D surveys of the coral reefs, taking environmental DNA, which gives us information about which metazoans are in there, and see whether that can, let's say the fingerprinting patterns of the environmental DNA can give us information about the functional diversity. So we first look at the biodiversity shifts just purely based on these 3D benthic surveys and orthomosaics, as well as eDNA, and then using various sensors, trying to understand the metabolic balance in these different systems. So areas that have a high cover of coral versus high cover of, let's say, soft coral, how do things shift? Do things shift with more autotrophs or heterotrophs or calcifiers and bioeroders? And
excuse me, I got some sign from the team keeper.
yeah, I'm. This is my
Doctor.
last slide.
Oh, okay.
Yes.
Thank you so much. Back to you.
Yeah, no, my apologies to everyone. I was told I was given an hour, and I just heard now that I have a half an hour. So I have to adjust things. I'm just ending here, and then I'll get to my last slide. So so ultimately where he wants to work is linking the metabolic balance with biodiversity, where you have sort of coral dominated and alternative states, and really looking at all these organisms we don't see but can catch in the eDNA and how they link to that. With that, I just want to go to my, hold on. I just want to go to my last slide with my email, and then I just want to quickly say thank you to my team. Are you seeing the full slide here?
Yes, clearly.
Okay, I just want to say thanks to my team with my lab, Tropical Marine Biodiversity Lab. This is part of the team. Everyone's distributed everywhere, so it's hard to get everyone in one picture. Also, a lot of the work in the field with different people. This is our work in Ambon, in Ambon in Maluku, and here in West Papua. So I just want to say thanks to everyone. This is the kind of work you can never do alone. It's very much a team effort of people on the ground and in the field. And thank you for listening.
All right. Thank you so much, Lisa, for your interesting presentation. Maybe you can stop your screen. Okay. Thank you so much. So now we will have a discussion session, but before that, we will have photo session, and I will let Mbak Dila to lead our photo session. Please, Mbak Dila.
Thank you, Pak
45
Rudi.
participants.
So let me see how many slides that we have. So we have 6 slides now. Let me start from the first slide first. Okay, and then second slide. Okay, got it. And then the third slide. Please give your best smile. Okay, and then the fourth slide. Smile. And then the fifth slide. Okay, and then the last slide. Okay, there you go. Back to you, Pak Dr. Ludi.
Thank you so much, Mbak Dila. Okay, so now we have a discussion session. So thank you so much, Lisa, for your time to share your expertise and experience. It's really give us really insightful about coral reef ecosystem under nutrient pollution. So for now, we have a discussion session for all of you who joining us online. So you can just click the raise hand icon. Yeah, we have here some people. I saw it. Mas Daski Rafid Abdunafi. You can just open your mic so we can hear your voice clearly.
Okay, hello everyone. My name is Anaf, and I'm currently a senior high school student in Depok. I would like to ask a question to Miss Lisa about the implications. So, provided that we already got the data regarding the different parameters of nutrients in the ocean, what are the implications that we can implement on assessing the ecological roles of each taxa in a particular coral reef ecosystem? Because we all know that coral reef environments are very delicate, and a skew in the balance of the nutrients surrounding the ecosystem can disrupt the ecological functions of each trophic level. So, is there any methodologies or like at least an implication on how we can use the data to assess the trajectory of an ecosystem on affected areas? Thank you.
Thanks. Yeah, very, very important question. So I would say at this stage, it's still very much in the very much developing the indicators and determining what are problematic values. So I think right now, I think what's key is to do the work to develop what are the thresholds, because I don't think we know that exactly. And so, and then indeed your question of like thresholds to what, right? So you have, I mean, you have to determine what effects they have. And as you indicate very correctly, um, increase in nutrients shifts the balance, and you have increase in other organisms. And that too, we see it's not so straightforward. So in the Caribbean and in Australia, where a lot of publications come from, there you see that the reefs are shifting to algae. But in Indonesia, it's not necessarily the case. We see that often reefs that are under a lot of nutrient pollution, they shift to um, soft corals. So I think finding out that question, what you're saying, of like which groups are actually then taking over, that first is a question. And then I would say the next step is identifying early indicators, because when you're already seeing the change in the coral reefs, then it's already quite late, right? So we want to try to see it before, so that you can then address the problem. So I don't know if that answers your question a little bit. I think we're still working on it, or there's still a lot of work for you to do.
Okay. It's clear, Mas Davi? Okay. Yeah. Then we move to the next question. Ibu Rini, you can open your mic. Yeah, Ibu Rini.
Thank you. Hello.
Yes, we can hear you.
Okay. Thank you, Professor Rudy. Good afternoon, Professor Lisa. Thank you very much for your insightful presentations. So as for your explanation before, these days there are some efforts worldwide and also Indonesia to enlarge their marine protected area, or as you know as MPA. So based on your experience, is there any standard for optimal area of MPA to be applied, but then functioning in the same time to support ecosystem sustainability and carrying capacity? Is it always linear, the connections? It means that is it a bigger area of MPA means better conservation results, considering the budget and maintenance efforts? And the second one is, do you have the recommendations on how we keep the core zone intact while providing the income or food for local community? Thank you.
Yeah, these are critical questions indeed, for MPAs. So so with respect to the size, I know, I mean, this is not work that I've done, but I know from other work there, yeah, there are some ideas about like the minimal size it should be, I think. I don't think it's necessarily linear, because exactly as you indicated, it all depends on the capacity. And so also some work that Agustin Capriati did is they evaluated like, okay, what is currently the capacity in these MPAs? And you see that formally from, let's say, the from the government, let's say, not many people actually work in MPAs. And so if you don't have a lot of people and they're also their skills are not that high, then it's very hard to manage the zones, keep it all going. So I think, look, you have to start somewhere. And I think it's great that Indonesia has decided, okay, we need to map the MPAs, we need to gazetteer them, we need to identify them. I think that's a great start. And then you have to start working on the quality of, like, the zoning and how to balance. The key thing is how to balance the economics, the local economies and the ecological benefits. And that only works by doing a lot of on-ground work with communities. And exactly how you ensure those core zones, that's something, that's a little bit more on the social science that I work on. But I would say if you want an effective MPA, it is key to consider, is there enough capacity? Are people trained well enough? Is there enough socialization and also buy-in that people, are part of it? Because as long as people don't feel part of the MPA, it's hard to keep it going. But I think, just to get back to the topics that we're working on, I think addressing the stressors and identifying which stressors are problematic, or which stressors are present per MPA, is a very important part of success. So first mapping the stressors and then identifying how they should be addressed, and then subsequently, doing all the other work as well. But I think that part sometimes gets forgotten. But thank you.
It is clear, Ibu Rini, the answer from Lisa.
One more thing, Dr. Lisa. Do you have like a specific protocol or standard maybe in Europe? Let's say if you want to provide or make a new MPA, let's say this amount of area, we need to provide this amount of resources or budgeted or person. Do you have something like that?
So to be honest, I don't know if that exists. There's, like I said, there's a paper we recently published with Agustin Capriati and a whole team that gives some beginning idea of what kind of staff capacity you need for your MPAs. But I don't know if it's quite, maybe answers your question entirely of this big, you need this many people. It also determines very much in what is your MPA, because I mean, as you know, Indonesia is huge, and so each MPA is very different ecologically, geomorphologically, socially. Some MPAs have lots of people living in it, and some MPAs not so many people. So it does depend on your MPA and, like, what are the issues, so I think there are a lot of parameters to take into account. But I don't personally know of so I know the IUCN has a general protocol, right? And as does the Indonesian government. But I think it is a bit of a process for each MPA.
Okay, I think it's enough from Ibu Rini. We can give another opportunity for other participants. Maybe someone can raise their hand.
There's
And
some
for
things there's also in the chat, I think.
Okay. Yeah, I found it from the chat. I will read a lot for you, Lisa, from Ibu Pasca. I'm curious, why did you link Indonesian Papua with ABC Islands, Caribbean, MPA's counterpart, given their coral richness index do not exactly match?
No, very good question. So what we are looking for within INREEF is we take case sites, right? So we have case studies in Indonesia as well as in the Dutch Caribbean, where we go into depth to really understand the systems, etc., etc. But then based on that, we want to take a step back to identify, are there general indicators or general patterns or feedbacks that we can identify in all systems, so that it can sort of inform, well, for example, for the questions before, inform MPA managers or policymakers of what are sort of general things that we should look at and indicators. So that's why it was for us quite important to take different types of systems that are under different development. So that's where that came from. But ultimately, absolutely correct. If you really look on the ground, they're very different systems. Yeah.
Yeah. Thank you, Lisa. Then for the next question from Taruna Mulia. Thanks for your enlightening presentation. Based on your presentation, most physicochemical or nutrients, indicators or variables so far only consider the absolute value of the concentration of each variable. In reality, however, interaction among these variables occur naturally. What are your views on the potential of using ratios of composite index such as nitrogen, phosphate, and silica, I think? The ratio as employed in the Index of Coastal Eutrophication Potential or ICEP. Additionally, based on your experience, is there a best example of how to assess the spatiotemporal aspect of such an index and its implication for coral reef ecosystem? Yeah, you can
Yeah.
answer
So
the
this is a
question.
very, very important point. And these indices, as far as I know, come very also strongly from a lot of work has been done in lakes and in rivers, right? And so based on that, we can get quite far. But where what we see right now in coastal systems is, yeah, these kinds of ratios are done for when you have very large scale things, right? Based on satellite imagery and these things. We wanted to be careful to just directly use that and translate that on the highly dynamic, closer to the coast, smaller scale systems. But that's absolutely where we want to go. So yeah, I didn't show that, but of course you can also, you absolutely have to work on these ratios. And they can give an indication, but we wanted to be careful with instantly starting on indices and first really look at the basic data and get a little bit of an idea, because it's within coral reef systems, how they react to NMP is a little bit different than a lot of other systems. So we wanted to get, so I think where we are now is understanding better, getting a better understanding of what is the temporal variation of these, and then indeed the indicators, the ratios, and then potentially the indicators. But I think the key point that this person identifies is, yeah, it's temporally very dynamic. And that's our issue right now, in that a lot of the monitoring, a lot of the work isn't that temporal. So it requires quite a lot of time and money to do. And I think we have to just sort of evaluate what is feasible. And that's why we're working with proxies. So we're also working with stable isotopes from macroalgae. We're working with microbial work. We're also looking into passive samplers. So we're looking at sort of solutions for the realities on the ground. But yeah, that it would be great to go in that direction. Yeah.
Okay, I think that's clear, Lisa. Then we move on the next question in the chat, also from Pak Indra Yanuar from BRIN. Thank you for the very interesting presentation, Lisa. Our research team have also found similar result to what you shared in several coral reef regions in Indonesia. The nutrient levels that our team found across Indonesian coral reef water were almost never below the average acceptable values reported in coral reef journal papers. Besides several areas from local anthropogenic pollutant, we also suspect that this different baseline is due to the trophic levels and the naturally different water fertility in Indonesia, which may consequently create a unique ecosystem that is distinct from location in subtropical region, in your opinion. what step can we do to establish an acceptable standard for this nutrient in Indonesia so that we can recommend it to policy makers? Wow.
Well,
Maybe you can.
first off, great that you've also
Yeah.
been doing this work. It would be great to talk more. So maybe you can also send me an email later to Indra Januar. And yeah, so yeah, as you find that there's this one standard threshold doesn't quite work for everywhere. So I think what's key is first to communicate clearly to the government that the goal should not be one value for all of Indonesia, and that the goal should probably be threshold indicators per region or per MPA. I mean, maybe you could set like an absolute maximum threshold for certain ecosystems. So for coral reefs, you have one, for seagrass, another. Maybe you can work with that. But what we're trying to do now with the local teams is try to set up in the next 2 years, basically, and this, so it would be great to work with you as well, to set up a sampling design where you sample the water quality and as well as these proxies in different areas that are under different exposure, as well as many reference sites, right? Because we need the reference sites in order to evaluate what are, let's say, normal or non-polluted areas. And based on that, we're hoping to get a bit of a sense of how dynamic is it and how does it shift according to the reference sites. And based on that, we're hoping to get some ideas, in any case, per MPA, but maybe we can get to some regional areas. But in all honesty, I don't know. I think we have to be very careful with setting values, because if you, before you understand the system, already set these values, then the numbers start taking a life of their own. The question is also, for a lot of the monitoring, the question is, maybe if you can't monitor frequently, maybe we shouldn't measure dissolved inorganic nutrients. Because if you can't do it frequently, maybe you just don't get enough information. Then maybe it's better to work with passive samplers or to work with proxies, for example, the nitrogen from algae or the microbial communities. So that's something else to consider. Yeah, maybe a less direct answer than what you would like, but I really value what you identify. Thank you.
Yeah. Good. Thank you, Lisa. Any more questions from the participants? And I also heard from you, Lisa, that you also will have another schedule, right? So,
Yeah, then maybe one more question.
yeah,
And then
maybe one more question, because Lisa will have another schedule or agenda, so she need to prepare for the next agenda. We will end it like 3 more minutes. So one more question, maybe. If there is no question, so maybe I can just close. Yeah, I think there is no question anymore. So thank you so much, Lisa Baking, for your time that you have shared your expertise and experience. So we hope that we can work together and collaborate. And then, yeah, the time for now, I give back to our MC, Mbak Dila, to close this session. Thank you so much, Lisa.
thank you so much.
Thank you, Dr. Ludi, for beautifully moderating the session. And thank you, Dr. Lisa Bekin, for such an eye-opening and deeply informative presentation. And then one key point that we need to consider as the closing remark today, as stated by Dr. Lisa Bekin, coral reef health is indeed a complex issue, but through outstanding global collaboration and scientific insights, such as those shared today, we can work towards more effective preservation strategies and ensure a sustainable future for this vital ecosystem. Okay, and we have now reached the end of the webinar. Distinguished guests, ladies and gentlemen, Bapak dan Ibu, The link for attendance list and a certificate evaluation form has been posted in the chat box. Please ensure that you fill the form before you leave the room. On behalf of the National Research and Innovation Agency, or the BRIN, we would like to express our deepest gratitude to our speakers, to our moderators, and all participants who stayed with us until the very end. Please stay connected with us through our social media, Instagram at EcologyBRIN, and our YouTube channel. So please, I'm so pleased to know that some colleagues have made some new networkings about coral reef research, and looking forward to see you again at our upcoming whatever opportunities to see you in other session. Stay safe, stay healthy, and have a wonderful day ahead. Thank you very much. Have a wonderful day, and wassalamualaikum warahmatullahi wabarakatuh.
Thank you again. Bye.
Thank
Thank
you,
you.
Lisa Bekin. Thank
Waalaikumsalam.
you,
Thank you very
Ludi.
much. See you again.
Thank
Thank you,
you.
Linda.
Okay, thank you, Miss Lisa.
Thank you. Assalamualaikum.
Waalaikumsalam.
Salam.
Silakan, Pak. Silakan.